Hi, I'm Glenn Gaudette from the Department of Biomedical Engineering at Worcester Polytechnic Institute, and I'll talk to you today about our scientific data management in biomedical engineering. What's involved in all the data acquisition? We start off 2 days before surgery, and we incubate our cells in quantum dots, what we call QD solution. The reason for this is we want to label our cells. And so we need to know what lot number of mesenchymal stem cells we're using. We can passage them multiple times, and so we grow them in culture dishes, and then we passage them. And so we want to know what passage number they are. And then we want to take a picture to confirm that the quantum dots have actually been taken up by the cells. When we take that, we have to take fluorescent images, and so we need to keep track of things like, was it a phase contrast image, was it a fluorescent image? If we're going to overlap two different types of images, we need to keep track of those images. The objective we used, we want to put a scale bar on the image, and if we've taken multiple images in the same well that we have the cells in. On day one before the surgery, day -1, we load these cells onto our biological suture. So we need to think about the concentration of the cells that we use in the solution, the volume that we deliver onto the cells, the volume that we deliver onto the suture, and then we want to take more images to confirm the cells are loaded on the suture. And so this here is an example of one image showing the cells in green and in blue loaded onto that fibrin suture. Again, are they fluorescent images? What kind of images? Are they just focused on the green channel, the red channel, the blue channel? Are they overlapped? What objective have we used? The scale bar. One of the other things we need to keep track of also is this suture here. What suture do we use? What size needle do we use? What batch of fibrinogen and thrombin do we use to make that suture with? And so we need to keep track of that in regards to the cells and in regards to the image for that specific experiment. When we get to surgery day, we start off pre-op, day 0, and we do things like weigh the animal. And so we want to know how much the animal weighs before we operate. We got to keep track of the drugs that we administer. We like to keep an eye on the heart rate and on the oxygen saturation in the animal's blood. And if we have extra cells, we'll fix those cells to make sure that we can keep an eye on the cells that we're delivering and see what state they're in when we deliver them. Intraoperatively, we take gross images of the heart and where the suture is placed. And so this here is an actual image from the operating room here. And this black line you see here is a suture, and so we want to know where in regards to the heart is that suture placed. Then when we deliver our cell-loaded biological sutures, we want to keep track of when they're delivered and what time they're delivered, where they're delivered. And so here in this image here, this pink thread-like device is our suture that we're sewing into the rat heart. We also need to keep track of what drugs we've administered and when we've administered them, what the heart rate is, and what the oxygen saturation is throughout the experiment. And then immediately post-op, we want to continue to track the heart rate and the oxygen saturation. We want to confirm that the cells loaded onto our extra sutures. And so we usually, if we implant one suture, we'll quite frequently bring in 4 sutures, and we'll take one of the extra sutures and fix it to confirm that the cells are still on that suture after the procedure. And so that's not a suture that is delivered to the heart, but that's kind of a control suture. And again, we're going to want to stain those and perhaps take fluorescent images. About a week after surgery, or post-op day 7, we want to start acquiring some data to look at the mechanical function in the heart. And so we take high-speed images as the heart contracts. We'll also take a gross image of the heart, and so that's this image here, looking at where the sutures are placed. We put a catheter, and if you look down here, you can see this white line. That's a left ventricular pressure catheter, and so we put that inside the left ventricle of the heart, and that measures the pressure. And so what you see down here is the pressure going up and down, and that's 1 heartbeat. We also want to record the oxygen saturation levels again, and then we take our high-speed images of the heart. And so when we take a picture of this heart here, in 1 heartbeat, we could take about 100 pictures of the heart. And I'll explain to you in a little while why we do that there. After we've acquired all our images, we'll take the heart out and we'll fix it, and then we'll process it. Before we fix it, we'll cut the heart right in the middle here between where the suture goes in and where the suture comes out. We'll call one of those sections the apex, this region down here, and the other the base, because this is the base of the heart and this is the apex of the heart. So now we have two separate sections. We've got to keep track of both of those sections. So we will also cut those sections up and keep track of the heart section it comes from, whether it's the apex or the base. When we cut them up, we need to keep track of which slide it is. Is it the first section off the heart, or is it the 10th section off the heart, or the 100th section off the heart? We'll take images of those heart sections. Sometimes we'll stain them to look at different proteins in the heart, and so we've got to keep track of those stains. Sometimes we'll take regular epifluorescent microscopy images. Other times we'll take confocal images. And so we've got to keep track of all those images, which section of the heart they were taken, which heart they were taken, and which experiment they're taken from. In regards to the mechanical function analysis, we look at displacement between two consecutive images. I'll go into this in a little bit more detail in a minute, but this means we look at displacement between two images, and if we take 100 images per heartbeat, we want to look at displacement over 100 images over one entire heart cycle. We also want to correlate the pressure with the displacement, and so we want to know what is the pressure at various points in the images. And this will allow us to determine displacement within small regions of the heart. In addition, we have written records, and so for example, here's an example of a preoperative medical record for one of our rats, where we keep track of the weight, the heart rate, the oxygen saturation, the drugs that we've delivered, the dosages, the lot number for the cells that we delivered, the mesenchymal stem cells, passage number, the time of the procedure, when did we make the incision, when did we deliver the cells, when did we close, what volume did we deliver, and so this is volume for drugs, how much suture did we deliver, what drugs did we administer, and again, what is the heart rate and what is the oxygen saturation. A typical data set for one experiment in regards to the raw acquired images, we could take 1,000 images per data set of the heart, and in a common experiment we'll take about 10 data sets, and so that's about 10,000 images that we'll take per experiment. In addition, these images are just the raw images of the heart. We also take an analog to digital input, and so we'll take that left ventricular pressure, for example, and we want to know what the left ventricular pressure is at each time we take one of these images. We also take the general images, which you see here on the right, and then we take our histological sections. We generally will take 3 sections and put them on a slide, and we'll take about 500, one heart will give us about 500 slides. In regards to the data collection for the high-speed images, in general, we'll take about 200 frames per second, and we want to get about 30 frames minimum per cardiac cycle. So per heartbeat, we want to take at least 30 frames. We take it at an 8-bit grayscale. We apply the silicon carbide nanoparticles with retroreflective beads. What that does is that it increases the light intensity distribution. The bottom line is our technique here essentially measures particles that are on the surface of the heart that we put there, and it measures where they move from one point to the next. So when we're acquiring these images, we want to save them first with the experiment number, then the data set number, the frame number, and then the file format. So for example, experiment number 0231, data set 2, and frame number 8 will be stored in this format here. The pressure data will be saved as experiment number, data set number, and then the file format. Because for each one of these images, the pressure is saved in this PDT file, which is similar to an Excel file. And so we store the pressure for an entire data set in one Excel file. And so, for example, this here might be EXP0231, data set 2. And so this here would contain the left ventricular pressure data that corresponds to these images. We save our data as TIFF files because it has higher resolution and provides us with more data rather than a JPEG file. The bad news associated with that is those TIFF files are generally larger, and so it requires more computational space than a JPEG. The PDT is a specific file format for the data acquisition software that we use. And so after we've acquired it as a PDT through the software system, we'll quite frequently put it out as an Excel file so that we can then use that there to correlate our images with. In regards to the data analysis we do, we essentially can determine displacement in the heart at each one of these green and red boxes. And the way we do that is through some Fourier transforms through this mathematical equation here. I'm not going to go into all the details. The bottom line is we end up what we call an impulse function, this blue spike you see here. And that corresponds, the distance that that spike is from the origin corresponds to the displacement in the X and the Y direction in the camera. And so along this axis and this axis here. That means for each one of these little points here, we can determine the displacement of that little point between two consecutive frames. We then move on to the next point. So if we start off with this red square, we then move to the green and red and green and so forth, and we can determine displacement at all these different locations between frame 1 and frame 2. Then we repeat that again for frame 2 to frame 3 and all the way up until the end of the data set. And again, that could be 1,000 frames. So the typical data sets for one experiment in regards to the analyzed data include these displacements in the X and Y direction. We call that U and V between two images. And so we'll have a summary sheet of that for each heartbeat, and that'll be a whole bunch, a series of U and V displacements for each image. And so again, a heartbeat will have at least 30 images in it. And so we'll have the U and V displacement for at least 30 images in that one summary sheet. From that, we can do regional analysis and look at how small regions on the heart are contracting. In addition, we'll take our histology images, and so we'll do things like hematoxylin and eosin, H&E. We'll do trichrome. We'll sometimes take phase contrast images. We'll save all these here on the computer. We'll do our immunohistochemistry. We may look at actinin, troponin, different calcium different cardiac markers, and we may take those images at different magnifications. Sometimes we'll combine those in a panorama image, and I'll show that in a minute. And we'll also take confocal images. The data naming format for our histology slides is first we note whether it's taken from the apex section or the basal section of the heart. And so if you recall, we cut the whole heart in half, and then we do our histology, our tissue sectioning from those two samples. So we'll give it our experiment number, A or B, and then the section number. So for example, 0231A, meaning that this is the section from the apex, and then the section number is 216. And so that means it's the 216th section that we've cut into that heart. Then when the sample is stained, we'll abbreviate for the type of stain that is used. For example, actinin, we add the underscore ACT. And so when we take the image, in this case here, which would be green, we would call that underscore ACT. One of the things we do with our histology images sometimes is we take a panorama view of the entire cross-section of the heart. What you see down here is an entire cross-section of the rat heart. This is the right ventricular cavity, this is the left ventricular cavity, and this here is the ventricular wall. And this here is the septum that separates the right and left side of the heart. We take higher resolution images like this here, and each one of these blue dots that you can see on this image here represent a cell. And then we take that image there and we combine it with all the other images to get a panorama view of the entire heart. And so this one image here may contain 30 or more individual images that we acquired one at a time. And so to name that, we'll call that again with the experiment number, the apex or base, and the type of stain. And then we'll add a row, which may be A, B, C, or D, and a column, number 1, 2, or 3. So for example, row A, column 1 is right here, row A, column 2, column 3, and so forth. And so this region here, for example, may be row D, column 3.